So, talking about microorganisms involved in bioremediation, there are aerobic bacteria, Pseudomonas, Alkaligenes, Sphingomonas, Rhodococcus, Mycobacterium. They have shown good promises for pesticides, hydrocarbons, alkanes, polyaromatics, and then methanotrophs. These are also aerobic bacteria, and they use actually CH4 methane for the derivation of carbon source and energy. Carbon source means they thrive on these methane molecules. They are methanogenic bacteria, and they are active against a wide range of compounds. For example, chlorinated aliphatics, trichloroethylene, dichloroethane, 1,2-dichloroethane, you can say. And there are anaerobic bacteria also involved, but anaerobic bacteria are not as frequently used because we are talking about bioremediation in environment. So if we are working in open air conditions, how can we provide anaerobic conditions? This is why anaerobic bacteria would be only restricted when we do bioremediation under controlled chambers, just like fermentation using a bioreactor or a fermenter. But still, there are some types of bioremediation where we can and we do use anaerobic bacteria. For example, the degradation of chloroforms in river sediments, as well as trichloroethanol, PCBs, polychlorinated biphenyls, and last but not least, fungi. They are able to degrade a very long, diverse range of persistent pollutants. Bioremediation have two strategies: in situ bioremediation and ex situ bioremediation. As the name suggests, in situ means at the site. In and ex situ is away from the site. In situ remediation is applied to eliminate the pollutants in contaminated soil and groundwaters. It is a superior method of cleaning for contaminants environment because it saves transportation cost and uses harmless microorganisms to eliminate the chemical contaminants. This feature increases the probability of bioremediation in close chemotactic affinity towards contaminants, and also the probability of bioremediation in points where bioremediants have not disturbed. This is a preferred method because it causes the least disruption of the contaminant area, and it is much of relevance either where the least investment and pollution are favored. For example, in some factories or in areas which are contaminated with dangerous pollutants. For example, in areas that are polluted with chemical or radioactive materials. Another advantage of in situ bioremediation is the feasibility of synchronous treatment of soil and groundwater. However, in situ bioremediation also possess some disadvantage. The method is really time consuming compared to other remedial methods, and it leads to a changed seasonal variation in the microbial activity because of the direct exposure to the variation in and uncontrolled environmental factors, and the use of additives may also lead to additional problems. And in the in situ bioremediation, we do the cleanup right at the site of contaminant. We know we don't need to remove any soils, we do not remove water, we just do the cleanup right at the site. You can also include some extra oxygen supply and nutrients so that the naturally occurring microorganisms can have a better environment for their thriving. And when they thrive, they will thrive with these pollutants, and with these physical and chemical favorable conditions through medium as well as oxygen will make them do the job in a much better way. The yield of bioremediation is actually determined by the kind of waste. Namely, if waste could provide the required nutrients and energy, then the microorganisms would be able to bioremediate it. However, in the absence of favorable wastes, the loss of bioactivity may be compensated through stimulation of native microorganisms, as I discussed earlier, by supplying oxygen and other compounds. In terms of this, in situ bioremediation is divided into two types based on the origin of microorganisms applied as bioremediants. So engineered bioremediation, which means intentional changes, and intrinsic bioremediation, you do not do nothing, simply allow the bioremediation to occur under natural circumstances, under natural conditions. In intrinsic bioremediation uses microorganisms already present. We do not do anything. We change nothing. We have zero human intervention in this type of bioremediation. And since it is somehow going on its own, that's why it's the cheapest. We are not doing anything. We're just waiting. We're just waiting for it so that everything is done by the microorganisms. Intrinsic bioremediation, in this is carried out without direct microbial amendment and through intermediation in ecological conditions of the contaminated region and fortification of the natural population. When sometimes intrinsic bioremediation isn't feasible, we turn towards the engineered bioremediation. And this is the second approach. And in this type of bioremediation, we do things like in situ bioremediation, as we said, we do nothing. But engineered in situ bioremediation, this type of bioremediation is performed through the introduction of certain microorganisms to a contamination site. As the conditions of contamination sites are most often favorable for the establishment and bioactivity of the exogenously amended microorganisms. Therefore, just like intrinsic bioremediation, the environment is modified in a way so that improved physicochemical conditions are provided. Oxygen, electron acceptors, and nutrients. For example, nitrogenous sources, phosphorus containing sources, they are required and they are given, supplied to enhance, to kick off, or to make the microbial growth better. Now we go to the next step, which is ex situ engineered bioremediation strategy. Now, if you see here, we say two types of bioremediation. Number one is in situ and the other one is ex situ. Then we say in situ has two types, engineered bioremediation and intrinsic. But turns out ex situ itself is one of the types of engineered bioremediation. The process of bioremediation here, in case of ex situ bioremediation, it takes place somewhere out from the contamination site, and it therefore requires transportation of contaminant soils or pumping of the water, the groundwater, to the site of remediation, which means we are not working at the site of the remediation, or we are not working at the site of the pollutant or the site of contamination. We have to take the soil, just like you see there, with the contaminated soil, we have to take it out and we have to transport it, and we have to do the bioremediation in some other places. Depending on the state of the contaminant, ex situ bioremediation can be classified as solid phase system, which include the land treatment and soil piles. The system is used in order to bioremediate organic wastes and problematic domestic and industrial wastes such as sewage sludge, municipal solid wastes. And this process, the solid soil phase, it includes three processes, including land filling, soil biofilling, and composting acids. So then the second type of ex situ bioremediation is called slurry phase system or slurry phase bioremediation. It includes solid liquid suspension in bioreactors, just like the fermentation processes. Now we have a controlled environment. All the bioremediation is going on inside a bioreactor or fermenter. The slurry phase bioremediation is relatively more rapid process compared to the other treatment processes. The contaminated soil is mixed with water and other additive in a large tank, which we call bioreactor, or we can call it fermenter. And then it is intermingled to bring the indigenous microorganisms in close contact with contaminants, soil contaminants, water contaminants, nutrients, oxygen, and other important ingredients are affected, and the conditions inside this closed chamber, which is a bioreactor, are regulated, adjusted, and controlled so that the optimum environment for the microbial bioremediation is intact, is prevailing. And after the completion of the process, the water is removed and the soil wastes are disposed of or processed to become to decontaminate the remaining pollutants. So the difference between ex situ and in situ bioremediation is, in case of ex situ bioremediation, you do not work at the site of the contamination. Rather, you take the contaminated soil or water and you treat it under somehow controlled environment. The next topic is phytoremediation. Not the next topic, but the next part. As the name suggests, phyto means plants, so it's the use of living green plants for in situ risk reduction and the removal of contaminants. And the plants have the capability to capture things from soil and from water to the root system. And when they take these pollutants, they take it above soil level in their branches and their leaves and possibly in their fruits. As I discussed, so they uptake the metal contaminants from the soil and translocate them to their above soil tissues. And once we have the plants totally grown and they have absorbed the metal pollutions, we cut the plants and we dispose them safely and we dispose them with care. Sometimes the contamination is so much, the severity of contamination, scope of some, how do you say, the sphere of contamination is so vast that you cannot do it just one time, so you have to repeat the process several times. A number of plants are used for this purpose. For example, the Avena species, the Brassica, I know it's not mentioned here, but alfalfa is very much used for this purpose. Populus trees are used for this purpose. Even eucalyptus and salix can be used. And most contaminants that could be removed are metal compounds, that include zinc, copper, nickel, for example. And how it is done, the degradation or the breakdown of this organic contaminants is actually done by the internal and external metabolic process of the plant. For example, enzymatic activity, oxygenases, these are the enzymes that convert, they degrade, they act upon hydrocarbons. Nitroreductases could be very effective in explosive degradations. And they are used in a number of reclamation, the phytoremediation. These plants are used for reclaiming a number of contaminated sites, such as ammunition wastes, chlorinated solvents, as well as the trichloroethane degradation. And also very important is for organic herbicides. Moving towards, and let's just discuss, we have discussed a lot about bioremediation is really good, it has blah, 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 all these good things, but there are some disadvantages of bioremediation also. It's a very slow process. The time it requires is not in minutes and hours, it's in days and months and sometimes years. You can never remove the heavy metals completely, there will still be some traces left. For in situ bioremediation, you must have soil which have high permeability. Otherwise, if you don't have high permeability, the extra bioaugmentation of oxygen and nutrients, it will not penetrate. Then you can never use bioremediation against all types of contaminants. There are restrictions. There are some exceptions where you cannot use bioremediation. Then the strains that we develop in our lab for bioremediation, actually in the food chain, protozoa feed upon them. So maybe we provide them for bioremediation to the contaminated soils, and there is a protozoan colony growing there, and they say, wow, food, and they just come and they eat all our bioremediation bacteria or fungi. And you can never always contact all compounds to be degraded. But there are a couple of more disadvantages. For example, scientific base is required for designing the process. It's not so easy to design the process because it needs a lot of expertise and a lot of pre-planning, which should be scientifically logical. And after this, we will step towards the next topic, which is bioleaching, which is somehow connected to bioremediation, but is not exactly bioremediation. There's still the involvement of bacteria in it.